Okay, today we're going to start our discussion about the cell cycle. Okay, the cell cycle. So today, one of our objectives is to be able to name parts of the cell cycle. You're going to hear about terms such as G1, meaning gap 1, G2, gap 2, synthesis, the S phase. You're going to hear about something we'll deal with next week called mitosis. Okay? And the last part of the cell cycle you're going to hear about is cytokinesis. So, those aspects of the cell cycle you are going to need to become familiar with. Okay? So, we're going to start the cell cycle today and continue into next week dealing with mitosis. So, why divide? Okay? The cell cycle, one of the big things that the cell cycle deals with is division. Why would a cell need to divide? Well, cell division prevents the cell from becoming too large. What's the big deal about having a large cell? Anybody remember? Jonathan? Huh? If it gets too big, it could burst. Okay? The cell membrane can only hold so much. What else? Okay? Remember when we did the diffusion of materials into the cell? That they couldn't get to the center of the cell if it were bigger. Okay? So, those smaller cells help to get the materials into the cell, as well as, I guess you could say, get it out of the cell too. Okay? And this is what we're looking at here. Just shows you, and it's really, really hard to see here, I know. I wish this slide, I'll probably end up replacing this. But we're looking at a small cell right here as opposed to a big cell. Okay, small, medium, large. And we're talking about the surface area to volume ratio. A higher surface area to volume ratio allows the cell to move materials in and out more effectively. Now, does that say, excuse me, does that say a bigger cell? No, it says a higher surface area to volume ratio. Where's the highest surface area to volume ratio? As they're bigger or as they're smaller? Smaller. Excellent. So smaller cells have higher surface to volume ratios. Is the way the cell reproduces so that you grow and heal certain injuries. That's another reason why we divide our cells. It is the way, division is the way that the cell reproduces and that we can heal up injuries, grow new cells. All right? Believe it or not, every one of us started off from a single zygote, a single cell, and then just kept dividing and dividing, dividing, until we became these multicellular organisms that we are. Okay? So if you look here, here's cells dividing, okay? And here's a damaged cell, okay? We'll talk about later on these damaged cells. Apoptosis, okay? Apoptosis we'll talk about later on, which is something about the death of the cell. So do we have cells in our bodies that die and no longer work. Absolutely. Okay? Absolutely. You know what? Our bodies can even recycle certain parts. Okay? Our body's got certain parts that their job is to look and say, hey, this cell is bad, but this mitochondria is still good. Can we use it on another cell somewhere else? Okay? Kind of like piecing together a car. Okay? A car might be bad, it might be in the junkyard, but are there still some good parts to it? Yeah. Yeah. Maybe you just busted off your rear view mirror. Could you go to a junkyard and find a rear view mirror? Yeah. I mean, you could go buy one too. But, some of those door mirrors are hard to match up. So if you find a car like yours, you can use that mirror. Same thing with cells. Same thing with cells. Maybe there's a damaged mitochondria that we could bring in another one there to help it out. Cells reproduce by a cycle of growing and dividing that we call the cell cycle. The cells reproduce by growing and dividing, what we call the cell cycle. Now here, I give you a little chart just to give you some background knowledge. Cell types and their lifespan. A skin cell has a lifespan of approximately 2 weeks. A red blood cell is approximately 4 months. A liver cell can be anywhere from 300 to 500 days. So that's about somewhere in the range of a year, right? How many days in a year? 365. So 365 falls in there. An intestine. The internal lining of your intestine. Those cells will last 4 to 5 days. That's not very long, is it? Okay. The muscle and other tissue, though, of an intestine can last up to 16 years, those cells. So we have a wide range of lifespan, so to speak, of cells. Interphase. Interphase is a stage in which the cell is growing. It's carrying out cellular functions, and it's also replicating. What's replicating mean? If I would replicate something, I would be doing what? Making a copy. So what you're going to see here is during this interphase, what is being replicated is the chromosomes. Okay? The DNA from the nucleus is being replicated. So this interphase is going to be a big, big part of the cell cycle. Right here, I have a picture of a cell in interphase and some big players here. The nucleolus, obviously. This circle here we know is the nucleus. We can see the nucleolus, and these little, not quite clear areas here is different chromatid inside the nucleus. We can see the plasma membrane. Here is the centrioles. Here are the centrioles. Okay, we have a pair here and a pair here, 2 pairs of centrioles. They are involved in mitosis, which we'll talk about next week. So we'll learn more about the centrioles. Did we talk about centrioles when we talked about the cell? Okay, look back in your interactive notebook. Let's check to see. I don't remember doing that, but let's turn to those cell pages where we had all those diagrams. I got yours, don't worry about it. Okay, where all those diagrams were on the cells. Here we are. Okay, I don't see centrioles yet. I don't see centrioles. All right, so this is going to be something of the cell that we haven't discussed yet, centrioles. Okay, they're a big player in the whole part of mitosis and meiosis. Stages of interphase. Okay? We have 3 different stages of interphase here. They call G1, gap 1 is what that stands for. You might want to write that in. Gap 1, G1, is the first stage of interphase. It's where growth in normal cell functions are occurring. The S stage, or the S phase, is the second stage of interphase. And when I think of S, I think of synthesis. Okay, S means the synthesis phase. The cell is copying the DNA in preparation for cell division. A sister chromatid is made and attached at the centromeres. The final stage of interphase is the G2, the gap 2 phase. The G2 phase is where the cell prepares for division of the nucleus. Okay, the cell prepares for the nucleus to divide. So the G1, the S, and the G2 phase in that order. Now, we talked about something here, a sister chromatid. Okay, this isn't in your notes, but you might want to draw it in somewhere where you got a little bit of room. Okay, I can tell you your cell cycle that you're going to put below it is this big. Okay, now a sister chromatid. What is a sister chromatid? Okay, if you look, right here are what we call homologous chromosomes. They are chromosomes that match up, and we have a centromere, something in the center. Now, you can see in this purple one, look at this X right here. It looks like an X, doesn't it, with a centromere? What that is when we were back here talking about this S phase, where the cell copies the DNA, well, that is the copy. The one purple chromosome has copied itself, and it's attached at the centromere, and we call these 2 sister chromatids. Okay? Sister chromatids. So a sister chromatid is a duplicated chromosome that is identical because it's duplicated. Now, I don't know if we'll talk about this. We'll probably talk about it more in meiosis. Something called crossing over will happen. Okay? But these sister chromatid are duplicated copies of a single chromosome. All right, now that you got your sister chromatid colored in there or in your interactive notebook, let's keep going on. Now, this is going to be, once I push this up, this is going to be the diagram of the cell cycle. I'm going to pass this out to you. This is the actual diagram that you will put in the bottom of page 88 there. So this is what you need to cut out and put in there. Now, it says on the board, C, C, and P. What's that? C, C, and P. Any idea? How about cut, color, and paste? There you go. Cut, color, and paste. And what are you going to cut? what I just gave you. What are you going to color it? There you have it, folks. There's a diagram that's almost identical to yours, except for the fact that it's not colored, okay? And it's not labeled either, is it? You're going to need to label, okay? Yours has numbers, so you can either label it right on it, or you could write number 1 is, number 2 is, number 3 is, okay? But you need to cut, color, and paste this into your interactive notebook.